Hi everyone, let's begin with the last chapter of bacteriology. And first and foremost, if you reached up till here, you're definitely a strong one because it's quite tough to go through the entire bacteriology portion. In case you finished it, you've reached this chapter, congratulations to you on that. I hope I've been able to make this journey of bacteriology a little more simpler for you, if to say the least. And yes, we're going to start with some miscellaneous bacteria now. So my first thing that I was doing up till now was that MRCS. If you remember, we've done the spirochetes, we've done chlamydia and rickettsia. Now we are left with M for mycoplasma. So mycoplasma is something known as the Eaton's agent. And before I go into the details of mycoplasma, I'm actually going to explain mycoplasma from the name mycoplasma, because now 90% of the information will be covered here. So what do I say? Mycoplasma, MY means M. It is the most common cause of walking pneumonia. And walking pneumonia is what kind of pneumonia? Guys, I've taught you pneumonia in your pathology lectures also. Pneumonia is a kind of this walking pneumonia is a kind of an atypical pneumonia. So can I say this is also the most common cause of an atypical community acquired pneumonia, not community acquired pneumonia. See, look at the difference. The most common cause of community acquired pneumonia would be Streptococcus pneumoniae, but the most common cause amongst the atypical bacteria causing it is going to be mycoplasma. Now coming to CO is going to be for cold. It shows you a cold agglutination test. It is cold agglutination test positive. Also, please note the C and the C will remind you of something I taught you in the beginning in general micro. Number one, it is going to not have a cell wall deficient form. And number two, is there something in the cell membrane? Yes, the cell membrane has sterols, which other bacteria don't. Remember, I told you, go back to your general microbiology lectures. I've taught you that the cell membrane doesn't have sterols, but here it has sterols. So remember, a cell wall is not there and cell membrane has sterols. Now we come to the next thing, PLA. I'm going to convert it into PPLO, because do you know this is also known as PPLO? This organism is given another name called as PPLO. What is PPLO? It is a pleuropneumonia-like organism. Pleuropneumonia-like organism, right? PPLO. That is the reason that now if someone asks you what is the agar, you will say it is grown on a PPLO agar and it shows you the fried egg colonies. So it's a PPLO agar and it shows you fried egg colony. Simple. Now we come to SMA. Firstly, S, they will ask you what is the stain. The stain is a stain called the Dines stain. I'll be showing you a picture. And also, I want to put up SMA together. SMA tells you this is the smallest bacteria. This is the smallest bacteria that we have. So simple. Now everything, I told you, 90% information is sorted. Jaldi se revise. My tells me it is the most common cause of walking pneumonia and atypical community acquired pneumonia. CO tells me cold agglutination test positive. It does not have a cell wall and its cell membrane has sterols. PLA tells me it's known as PPLO, pleuropneumonia-like organism, and it is PPLO agar showing you fried egg colonies. I put a stain, yes, that is known as the Dines stain, and SMA stands for smallest. So Mycoplasma, Eaton's agent, is sorted. Now let's go on to the details. Is this the smallest bacteria? Yes, SMA is the smallest bacteria. It's somewhere in the nanometer range, which probably the viruses are. So it's definitely the smallest bacteria. And it's so small that easily, if you try to filter it out, it will pass through the filters very easily. Next, under the C, I have told you it does not have a cell wall and its cell membrane is made up of sterols. I already know it. Coming to the next, what is it going to be grown on? It is also known as pleuropneumonia-like organism. It is grown on a PPLO agar. So what exactly is a PPLO agar? Most important thing that you have to know that it contains penicillium. It contains penicillium and thallium acetate, because that is how mycoplasma will be grown and others will not be grown. These are the selecting agents. So minimum भी याद रखना है तो no, PPLO contains penicillium and thallium acetate. And now if you see what kind of colonies do you get on PPLO agar, we get the very, very famous fried egg colonies. We get the fried egg colonies. So I can very well see over here, if I zoom in, you can see that there's a halo around, there's a dense center and a halo around it. Hai na? So these are the fried egg colonies. But you're saying, ma'am, these actually, it's such a small little organism. So the colonies are also very, very small. You might not be able to see it. You will say, ma'am, I'll add one drop. I'll add one drop of what stain? on top of them. So remember, on the surface of the culture, I add a stain called the dyeing stain, and now can you look at those? The same thing, the dense, it has a very, very dark center like this, and then it has a lighter area around it. That is how your fried egg colony is going to look like. Of course, you have so many fried eggs that you have, which I'll talk to you in a while. But yeah, dyeing stain I already knew. Now tell me, what was the test that I told you? For mycoplasma, I told you it is a cold agglutination test. Go to your immunology chapter, you know it's a heterophile agglutination test, but all these testings are all non-specific. The most or the best test, nothing can beat genetics. Nucleic acid amplification testing, polymerase chain reaction, that of course is going to be the most sensitive. That is a previous year question. All said and done. This will be like, you say, ma'am, ye to mycoplasma word you have explained. Exactly, can you tell me what are the different diseases that it causes? Yes, like we said, it is the most common cause of walking pneumonia. Number one, walking pneumonia means patient is going to walk into your OPD. He's not bedridden, he's not in the ICU. And is it the most common cause of community acquired pneumonia? No, the most common cause of community acquired pneumonia was Streptococcus pneumonia. This is the most common cause of community acquired atypical pneumonia. Done. These two I have already known. Guys, do I know something else also? Is it associated with some autoimmune hemolytic anemia? Socho, the cold autoimmune hemolytic anemia known as cold agglutinin disease. I have taught you this in your pathology lectures. The cold agglutinin disease definitely is showing association with mycoplasma. I gave you a mnemonic also. Cold agglutinin disease was because of IgM antibodies, and with IgM I had made you learn that mycoplasma leads to it. But now you mean to say only and only lung to nahi hai, other things are also there. Can there be extrapulmonary? Yes, there can be a lot of other systems involved. The heart can be affected, brain can be affected, Guillain-Barré syndrome is known. So there are a lot of other features. Like I said, the meningitis, encephalitis, Guillain-Barré syndrome, the heart can be affected. It can cause pericarditis. Guys, it can also cause Stevens-Johnson syndrome. So definitely the heart, brain, skin, there are other organs also affected. Extrapulmonary manifestations are very well known. And what is finally going to be the drug of choice? The drug of choice are going to be the macrolide. So overall, as I go back to my mnemonic, mycoplasma pneumonia ke liye mycoplasma yaad kara, so your life becomes simple. Let's go on to the other miscellaneous bacteria that we have, and they are going to be the bacters. When I say bacters, you know we are talking about the Campylobacter and the Helicobacter. There's Campylobacter and there is Helicobacter. So all of these bacters are said to be COPS. What is COPS? They are catalase and oxidase positive. What did I say? COPS means that they are catalase and oxidase positive. Number one. Apart from that, what is the S standing for? All of them are spiral rods. They are spiral rods. So whenever I use the word a bacter, Campylobacter or Helicobacter, you need to know that they are catalase oxidase positive and they are spiral rods. So first I'm starting with Campylobacter, and I want all of you to learn like I'm telling you, like mycoplasma ka mnemonic tha mycoplasma. Campylobacter ka mnemonic hai Campylobacter jejuni. So that is only the mnemonic. So please note, when I'm talking about Campylobacter jejuni, first I'll tell you the mnemonic so that the rest of your story becomes a little easy. So jejuni reminds me of June ka mahina. June ka mahina, especially at a place where I live, that is Delhi, June means boiling hot temperature. So June is going to be reminding me of 42 degrees. Yes, it grows at a 42 degrees centigrade. So remember, Campylobacter jejuni grows in the June ka mahina, that is at 42 degrees. One part is done. Next you're going to say, okay, it's a bacter. This means, ma'am, I know this is going to be catalase oxidase positive and it's going to be a spiral rod. COPS is there. Now we come to camp. I want you to learn it as camp. Guys, when you go for camping, how many of you are fond of camping? I'll give you a glimpse. When I was in my MBBS, I remember I was really fond of camping. That time I used to go also, but now unfortunately or fortunately, I don't know, but life does not permit that much of time. And you might feel that, why ma'am, why don't you have time? Aapka to NEET PG sab ho gaya. So then that is what you feel. You feel that you will get time as you grow older, but that just doesn't happen. So it's not as frequent as what I think. Now I feel MBBS was only the best. So make use of this time. Enjoy this time. Okay, coming back. So what is it in camping that you are worried about? Number one, you are worried about, what are main concerns while going camping? Camping jane ke time, I am concerned, what am I going to eat? Where am I going to get the water from? A very genuine concern is, where will I go to the washroom? Loo, washroom. And of course, think of the good things. You look at this camping photo, all that you will be seeing, open sky, you're going to see a lot of stars. So yes, you're going to see stars. So काफी कुछ worry भी करना होता है. You have to worry about the food, you have to worry about the water, you have to worry about your washroom. So you can say that, ma'am, इतना worry करेंगे, so this is obviously going to result in इतना worry करोगे, तो it is going to result in some brain problem only. Too much of worry I have, food ka, water ka, washroom ka, fine. Also, please note, why am I telling you this food, water, loo story? Because now if I'm going to ask you, what are the causes of Campylobacter jejuni, or what does it cause via what route? So you will say that this is because of food. Food matlab, what did I say? Food. Food matlab, this is because of eating something garbar. That is most commonly contaminated poultry. So you went for camping and you ended up eating some random animal over there, contaminated poultry. So food ka to problem ho gaya. What is it going to do? I'm next worried about water and loo. So it's going to cause watery loo, means it's going to cause diarrhea, which is usually a kind of a watery diarrhea. It is usually a kind of a watery diarrhea. So of course, fever and pain to sabke saath hota hai. So clinically, it's going to cause watery loo, watery diarrhea. What is the incubation period? You will say, ma'am, mostly जो camping trips होते हैं ना, they are for approximately 1 week. 5 to 7 days का एक package होता है. So the incubation period is also going to be roughly the same. Camping का time 1 to 7 days. Next you say इतना worry करा 1 week के लिए, ma'am, worried about food, worried about water, worried about the washroom. The person ended up going half crazy. कुछ CNS complication हो सकता है? Yes, this can result in Guillain-Barré syndrome, which is by serotype O19. Okay, so you mean to say only this diarrhea wala problem is occurring? No, I can now say, I'll come back to this mnemonic. I can now say that only a part of it is intestinal. Intestinal matlab there is abdominal pain, there is watery diarrhea. In fact, they say that this abdominal pain can sometimes be so severe that it starts resembling appendicitis. Patient might feel, or you, the clinician, might feel that there is appendicitis. It's pain in that region. It's known as pseudoappendicitis. So abdominal pain, fever, and what? Watery diarrhea mostly. Rarely bloody diarrhea, but primarily watery diarrhea, and this usually tends to be self-limiting. But there is also, all this was Campylobacter jejuni. Please note, there is something known as Campylobacter fetus, and that can cause all septicemia features. That is bacteremia, meningitis. It can go in different organs. It can cause all sepsis wala features. That is more featuristic of Campylobacter fetus. Whatever I'm talking about, this diarrhea story is to do with Campylobacter jejuni. Now let's come down to how is it going to look like under the microscope. What did I say? They are catalase oxidase positive spiral rods. So that means that they are now going to be comma S or gull wing shaped. देखो या तो comma होगा या फिर उसका you can make an S out of it. So comma S or gull wing shaped, because we said they had to be something spiral, and we know we are anyway dealing with what? We are anyway dealing with gram negative organisms. So they are catalase oxidase positive. Spiral matlab they are comma S or gull wing shaped. Then you will remember I told you that you were seeing what in the sky when you go camping. What do you see? You see stars in the sky. So which kind of motility does it show you? It shows you the darting motility or the shooting star motility. The darting motility or the shooting star motility is noted. So now can you also make a list for me that shooting star motility was also shown where? Shooting star was also shown in Vibrio cholera. Over there I had taught you that not only Vibrio shows you this, also Campylobacter is going to show you this. Okay, so other than that, what will the stool sample show you? It will show you some pus cells. It will show you some pus cells, that is fecal leukocytes. So see, spirals I got to know the spiral shape, and stars I got to know the darting motility. Now finally coming to the media, guys. If the name of the organism is Campylobacter, then something Campybap is the media which I call it. It is most commonly what I use is Campybap. You can also call it the Skero or the Budzler media, and these are usually containing charcoal. They are charcoal based. The Campybap is very common. Campybap, Skero or Budzler media, and they are going to be for Campylobacter, and you will see these effuse droplet like colonies. So imagine you are camping in some really cold place up in the hills. So will you get that moist dew? Exactly. You are going to get effuse droplet like colonies, because now in this photo along with the stars on your tent I am also adding a little bit of dew drops. Okay, I'm also adding a dew drop so that you remember कि Campybap में I am going to get the effuse droplet-like colonies. Finally, what is the treatment? In most of the cases, you are only going to limit yourself to treating symptomatically fluid electrolyte, diarrhea, treat karo, that's it. Only in severe cases, or if the patient has extremely prolonged illness for many days, then you go in for azithromycin. So now I feel that your Campylobacter pneumonic and your Campylobacter is, or you can say Campylobacter ka mnemonic seems a little sorted. Now I can ask you, tell me what is food? It is because of ingestion. Camping gaye the to ma'am koi bhi meat kha liya, contaminated poultry. Water and loo matlab watery loo is going to be a concern. Watery diarrhea happens. In the stars, I'm looking at shooting star, darting motility, and itna sab worry karte I ended up having a complication called Guillain-Barré syndrome. If I talk about the camping ka trip, it was a 1 week trip, so the incubation period is going to be 1 week. If I now take the Campy out of it, I think we all know it is the Campylobacter sciro or the Budzler media, and what you get on Campylobacter is the effuse droplet-like colonies. What does Bacter tell you? It is catalase, oxidase positive, and spiral matlab it is going to be that S or gullwing shaped. Finally, Jejuni tells you June ka mahina, 42 degrees. Campylobacter sorted. The other bacter that we go on to is going to be the Helicobacter pylori, and so much about it I have told you. कि bacter है catalase, oxidase, positive, spiral, ठीक है. But this is also a Helicobacter, means it is urease positive. Go back to your bacteriology section. Have I taught you a mnemonic called punch kiss? I'm not going to repeat it because we've repeated it so many times till now. But if you remember, punch kiss mein I always used to tell you that H is going to be for Helicobacter pylori. So I can say it is coppus, if you can say that. That is going to be catalase, oxidase positive, also urease positive. ठीक है, वो extra है. What are the virulence factors that it has? This I've taught you in pathology also. CAG A and VAC A. What is CAG A and VAC A? VAC A matlab vacuolating toxin A, and CAG A means cytotoxin associated gene A. तो एक वैक्यूलेशन के लिए, एक साइटोटॉक्सिसिटी के लिए. साइटोटॉक्सिसिटी gonna cyto cell, toxic break, gonna break the cell by the type 4 secretion system. It encodes for something called the type 4 secretion system. I don't know how many of you remember, if you read too much of detailing of micro, if you feel, Shigella mein what was being used was type 3 secretion system. Shigella में type 3 secretion system था, but not very important information. Helicobacter pylori, CAG A and VAC A, these are PYQs in which they ask you that how does H. pylori cause cancer in the stomach, via which factors? So what are its virulence factors? Helicobacter के लिए CAG A and VAC A. What did I say? How does it cause stomach cancer? You are gradually going to ask me. This requires an acidic pH. Ma'am, acidic pH तो सिर्फ stomach में मिलेगा, but it has to go into the stomach. How is it going to go into the stomach? Of course, someone's going to eat it, fecal-oral, and who is going to get this kind of an infection from contaminated food and water? Someone who's living in poor hygienic conditions, someone जो overcrowding में रहेगा, उसे होगा. So obviously this is going to be the case. And you say that acid, सबसे ज़्यादा acid, who releases HCl? Antrum. What is the site in which H. pylori is going to go? It's going to go in the antrum of the stomach. So once it goes into the stomach, what are the diseases that it causes? So I'll draw 4 stomachs over here, okay? Because it is going to be associated with 4 diseases. The first disease is nothing but a gastritis. And let me ask you, which chronic gastritis does it cause? Chronic gastritis A or chronic gastritis B? It is B. If you remember, A wala chronic gastritis was autoimmune. B wala chronic gastritis was bacteria wala. So Helicobacter pylori is a bacteria. It causes chronic gastritis B. That chronic gastritis changes into peptic ulcer disease. So ये दोनों तो inflammation related things हो गए. If I ask you what are the cancer related things, so number 1, Helicobacter pylori causes adenocarcinoma in the stomach. And secondly, it causes maltoma in the stomach. Guys, what is maltoma? Maltoma is a lymphoma. And what kind of a lymphoma? It is going to be a marginal zone lymphoma. It is going to be a marginal zone lymphoma. So repeating once again, we have gastritis and ulcers and 2 cancers. Adenocarcinoma stomach, maltoma हमें पता है from pathology, it's a marginal zone lymphoma. All of them are associated with Helicobacter pylori, but I have just made Helicobacter pylori into one matlab deadly thing. It is only and only like villain bana diya. No, it's not always villain. Can it be protective also against some things? Yes, it is ABC analysis के लिए protective. Matlab A for adenocarcinoma of the esophagus. स्टमक के लिए इट माइट बी वेरी बैड, बट ईसोफेगस के लिए अच्छा है. इट प्रिवेंट्स अडीनोकार्सिनोमा ईसोफेगस. व्हाट इज द प्रिकर्सर ऑफ अडीनोकार्सिनोमा? Esophagus, Barrett's esophagus. This means it's protective against Barrett's esophagus also. And why is Barrett's esophagus happening? It is happening because of GERD. This means it is protective against GERD. If it is protecting, H. pylori is protecting, basically it's going to affect the acidity. Now, so it will not let GERD occur. If GERD nahi hoga, so the Barrett's esophagus will not occur. And if Barrett's will not occur, so adenocarcinoma esophagus will not occur. Nowadays, they also say it is having a protective role against asthma. So ABG analysis was made because of it. I don't think you need a mnemonic. I think you've understood it anyway. Finally, let's come to the lab diagnosis. We are going to divide the lab diagnosis. See, it's in the stomach now. So either you can do something from the stomach, so ये थोड़ा invasion होगा. Either there's going to be an invasive test, or you will be doing some stool wala non-invasive test. So under the invasive tests, number one, you can do a biopsy. From which part of the stomach will you want to do? You would want to go into the antrum. What is it going to look on microscopy? So it is a gram negative, and I have told you Kopp's mnemonic se pata hi lag gaya. It is a spiral shaped organism. It is a spiral gram negative organism. But you say that, okay, when you did a biopsy and when you saw the organism, why don't you use a couple of stains? Yes, we all know that whenever, see, look at this, this is a stomach biopsy and the Helicobacter pylori has started looking black in all of this. Helicobacter pylori has started looking black. So please note that this is referred to as the Worden Starry Silver Stain, WSSS. Remember, the Worden Starry Silver Stain is what is going to be giving you silver, matlab black color, is what is going to be giving you the black color in case of Helicobacter pylori, in case of Helicobacter pylori. So this is one, that is the Worden Starry Silver Stain. There's another silver stain known as the Steiner stain. Photo ya kuch puchenge nahi, but just pad lo once. That's another silver stain. But you know the problem is, for you to use these silver stains and then to get a black color is a little time consuming. Yes, there is something known as a modified Giemsa stain. There is a modified Giemsa stain, and you can see that the modified Giemsa stain has given that a blue color over here. Okay? Everyone can appreciate. So best to of course still WSSS, Worden Starry Silver Stain. Now that you've read bacteriology, matlab now you will feel that till now ma'am was so sweet and kind to us. Suddenly she's turning villainous because I'm asking you questions from anywhere now. Now I can ask you, I have that upper hand, I've taught you everything. Worden Starry Silver Stain was number one used for here H. pylori. It's two more places, but not important. Ek aur to you have to know. If you remember Bartonella Hensley. Bartonella Hensley was also something that was having this particular Helicobacter, the same stain as H. pylori, that is Worden Starry Silver Stain. So coming back, these were invasive tests. You took a biopsy, you did the microscope, you did all the stainings, okay. But if you don't want to be invasive, then what are all the non-invasive tests? Non-invasive में क्या क्या याद आता है? मैम एंटीजन देख लो, एंटीबॉडी देख लो, ठीक है. तो फर्स्टली अगर एंटीबॉडी देखना है तो you will have to do a ELISA. Antigen ke liye you can do coproantigen. There is also a breath test that we do, which is pretty sensitive, because I told you that Helicobacter pylori is a urease positive organism. It has an enzyme called urease, so you can do a urea breath test, which is quite a sensitive test. What else? There's urea test, or you can say the urease test. Urease test is also एक अलग से भी urease test you can do, that is known as the CLO test, because you know earlier they used to just say Bacter, Campylobacter and Helicobacter same होता था. Then they realized नहीं Campylobacter अलग, Helicobacter अलग. But till date they confuse it. They still consider it of the same family. They say it is a Campylobacter-like organism. Helicobacter को बोलते हैं Campylobacter-like organism. So when they say they have done a CLO test, मतलब nothing, they have done it for Helicobacter, it is a urease test. So what all are the biochemical tests, वो तो पता है. Catalase oxidase positivity तो पता था. Now I just added the urease for it, and urease के लिए कौन सा वाला reaction? The CLO test. What is going to be specific? Of course, the culture is going to be specific. You can use the same cultures as Campi. Campi वाले culture, Skero, Budzler, वो use कर सकते हो, because both of them are Bacter same family ना. तो culture use हो सकता है आपस में. Also, I want you to answer one thing for me. Do you remember the treatment protocol, triple drug and 4-drug regime? Only from pharmacology, I've got you something from the pharmacology lectures. We all know that there is a OCA regime, OCA regime is omeprazole, clarithromycin and amoxicillin. Then we have the OBM regime, so OCA and OBM. OBM may be, O is going to be omeprazole, bismuth salicylate and M for metronidazole. And then we have the OBTM. OBM to same hai, omeprazole, bismuth and metronidazole. The T is going to be the tetracycline. So we have the OCA, we have the OBM and OB tetracycline M, metronidazole. These are the ones usually for 14 days they are given. So that wraps up what? I think by now mycoplasma ho gaya, mycoplasma ka mnemonic, mycoplasma. Campylobacter kara, campylobacter ka mnemonic, campylobacter. Helicobacter I've already told you. Now I move on to the next one, which is a very short 5-minute thing. The next organism is Gardnerella vaginalis, which anyway tells me that this is going to cause a vaginal infection, bacterial vaginosis, common sense. This causes bacterial vaginosis, for which what will happen? The patient will have a discharge. Patient will have a discharge per vaginum. So I will say, fine, I will do a pap smear. I will take the discharge, and what do I see on a pap smear? I can see, देखो पहले तो let me take you back to pathology. If this is a pap smear, ये pink वाला cell कौन सा होता है? On a pap smear, pink वाला cell is the superficial cell. Pink वाला cell is the superficial. देखो superficial cell is pink वाला cell that you have, right? However, now on that pink wala cell, I am seeing on top of that pink cell, there are a lot of ऐसा blue color bacteria. Bacteria have stuck. What are these? These are not, मतलब bacteria तो है, but क्या type का bacteria है? These are nothing but cocobacilli. They are nothing but cocobacilli. So please remember, the cocobacilli are stuck on the cells. What do you call this? They are giving you a clue. They are giving you a clue that clue cells are gonna be present in Gardner. They will give you a clue. अच्छा ये दिख रहा है? This must be bacterial vaginosis. They are giving me a clue to it. In fact, please note that there is a very famous criteria. दो criteria है for Gardnerella vaginalis, correlating this with your gynecology. Amsel's criteria, Nugent criteria. What is Amsel's criteria? They say first and foremost the pH should be above 4.5. Then there is a test called the whiff test. What is whiff test? They say that if you treat the discharge with 10% KOH, there is a very whiff, means whiff fishy, whiff odor, whiff fishy odor or fishy smell is gonna come. And finally, the third one, you have seen that it shows you the presence of these clue cells. Can everyone see how the cell is studded with those ... cocobacilli dikh raha hai na? So repeating once again, there is a pH, there is a whiff test, whiff matlab fishy, and there is a clue cell. Ye tha Amsel's criteria. You also remember there's another criteria called Nugent's criteria, and now I have learnt it is new generation. Old generation replaced by new generation. Matlab, what is the usual old generation of the vaginal tract? The old generation of the vaginal tract is lactobacillus. This is normal, commensal, ye to hota hi hai. The old generation is lactobacillus, which is also known as what? Lactobacillus is also known as the Dodderlein's bacillus. Lactobacillus is also known as the Dodderlein bacillus, but now the new generation is gonna come. The old lactobacillus will be replaced by either Gardnerella, even Mobiluncus. Mobiluncus se bhi replacement ho sakta hai. Okay? So basically the new generation is coming in and old wala is getting replaced. That is the Nugent's criteria. How are you going to do the treatment of this? The treatment of this is going to be with the help of metronidazole. That is your final thing towards Gardnerella vaginalis. Chota sa bacteria, this is also done. One more chota set of bacteria that I'm going to take you towards is rat bite fever. With, I know, because it's an end topic by now, you're like half almost given up on life and a little, a little on the verge of a breakdown, because that is how micro is. So I've given you chota interesting stories. Rat bite fever. This much I've understood, it's going to happen with the rat bite. Two organisms, Streptobacillus moniliformis and Spirillum minus. So you will say, what are the diseases that it causes? So moniliformis sounds like Mona Lisa. Mona Lisa wants to go on a trip. Mona Lisa is wanting to have a trip to the hills. Mona Lisa wants to go on a trip to the hills, have her hill fever. Okay, so remember, have her hill fever is caused by moniliformis. So Mona Lisa going to the hills. Whereas what is this? Spirillum minus. Ma'am, minus plus ही करना है तो सुडोकू जैसा लग रहा है. Minus plus करना है तो सुडोकू, so it causes sudoku fever. Is this okay? So Streptobacillus moniliformis, so Mona Lisa is going to be Haverhill fever, and Spirillum minus is going to be Sudoku fever. अब होता क्या है? Because both of them are happening with a rat bite. So if I say that what is the difference between ये वाला rat bite fever and ये वाला rat bite fever, so you will say if the rat bites, is there something at the bite? That is only in spirillum minus. If they say there is a local ulcer and a lymphadenopathy, local जहाँ पे bite हुआ है वहाँ पे problem आएगा, that is going to be in spirillum minus, because in Streptobacillus moniliformis the local things are not there, but there is relapsing fever with rashes and arthralgia. So remember, the rat bite fever is going to be rat wala fever, that is relapsing fever. ठीक है, rat wala fever, and that is going to be relapsing fever. So relapsing fever is going to be seen here also, going to be seen here also. दोनों में rat bite है, दोनों में relapsing fever है, but who has the local symptoms of an ulcer? The spirillum minus is going to have the local symptoms of an ulcer. Okay, now let's talk about the other names. Please remember this Haverhill fever. Let's break it. It is erythemica. Erythemica matlab, ma'am, there is some kind of erythemica, means a reddish rash. Arthriticum. Arthriticum means there is some kind of arthralgia, matlab there is a joint involvement. And epidemicum, because it spreads like that. So the Haverhill, तो hills में epidemic हो रहा है, तो मोनालिसा को hills में जाना है, but hills में तो epidemic हो रहा है. ठीक है, कैसा कैसा stories we have to make in order to sail our boat through microbiology. तो मोनालिसा तो wants to go to the have her trip in the hills, तो moniliformis is gonna have Haverhill fever, but hills में तो हो रहा है the epidemic. So it is known as the erythemica arthriticum epidemica. And now you've understood na, दोनों में relapsing fever and rash and arthralgia है, but who has the local symptoms? The local symptoms are in spirillum minus. What is the incubation period? I know it's a little difficult to learn, but एक का है roughly 1 week and एक का है 1 to 3 weeks. ठीक है, now we come to the drug of choice, which is going to be the same for both, that is penicillin. That is what you have to learn for rat bite fever. This one is also done. Should we move on to the next one? A single slide that you have to know for Pasteurella multocida. What is Pasteurella multocida? ये सब gram negative चल रहा है. So this is something which is associated with cat and dog bite. So cat and dog say so many things. Dog bite say to of course dog bite nahi, but just look at dog. Dogs I can of course always think of rabies. Kahin nahi gaya. That is there. But other than that, do you remember any parasite which can be associated with dog? What is known as the dog echinococcus? कौन सा वाला था? Echinococcus. I told you na, dog tape from echinococcus. Yes, so that is also dog. So Pasteurella multocida is also associated with a dog and a cat bite. Okay, that is one thing. So now just go imagine a dog or a cat bite हुआ, तो local side पे तो inflammation होगा. ठीक है, for example knee में ऐसा bite हुआ, तो local side पे inflammation होगा, tenderness होगा, pus होगा. Then उसके नीचे there is joint and bone ना. So after that local side there is going to be joint में tenosynovitis, bone में there is going to be osteomyelitis. So local site होगा, then it's gonna spread resulting in tenosynovitis and osteomyelitis. Further spread हो जाएगा मैम, तो it is gonna result in septicemia. And finally the treatment is gonna be by beta-lactams. But that's all that you had to learn. So Pasteurella के लिए remember this is associated with some kind of a dog bite. जहाँ dog bite हुआ वहाँ tenderness होगा. After that the bone and joint will have a problem. After that sepsis, that is gonna be fatal. Done. Next one, I'm sure the next organism, these are rapid one slide organisms, might get you one out of all of them, you might get one question. So just quickly flip through it. The next one is known as Legionella pneumophila. Pneumophila has told me कि अच्छा कुछ तो pneumo मतलब there is some kind of pulmonary involvement, but it has two forms. Either it can be pulmonary or it can be non-pulmonary. So there is a pneumonic form and there is a non-pneumonic form. And you know guys, na, what is my favorite mnemonic in Path and Micro? P is not for P. So if I say Pontac fever क्या है? You say Pontac fever is not pulmonary, not pneumonic form. The Legionnaire's disease is the pneumonic form and Pontac P के साथ P नहीं, it's not pneumonic form. P is not for P. So then what is Pontac fever? So this much I've understood that Legionnaire's disease is the pneumonic form, pneumonia होगा. Pontac fever में क्या होगा? It is going to be a patient who's having mild fever. There is mild fever that the patient has. Apart from that, there is myalgia. There can be an element of non-productive cough. there can be an element of non-productive cough, and after that, yes, there is an involvement of the respiratory tract, then respiratory failure and death can occur. So is the primary complaint going to be the pneumonia wala features? No, it's a gradual thing that develops, that is contact fever. But this is very commonly seen how pneumonia, मतलब lungs में ही जाना है, तो it is going to be by the inhalation of aerosols, especially from air conditioners. So air conditioners के साथ Legionella is something that is associated. Not only that, people who are prolonged hospitalization, who are in ICUs for a very long time, or have less of immunity, or have harmed themselves with smoking and alcohol, those are the people who are going to have Legionella. So repeating, abhi we also have to do the different pathogenesis and the culture media, that is also important. But please note, Legionella pneumophila से ultimately I realized या तो direct Legionnaire's disease होगा जो pneumonia है, or there is a contact fever which will gradually result in lung involvement. Either ways, lungs में तो there is going to be, it's a black day for the lungs. What am I saying? It's going to be a black day for the lungs, which means it's going to go directly to the lungs. If they ask you, this bacteria goes in the alveoli, तो alveoli के कौन से cells में is it going to multiply? It is going to go, and which is that hungry cell which is going to eat it up? It is always going to go and multiply in the macrophages. Then it is going to disseminate via the blood and the lymphatics. Then it will say, okay, enough of the pulmonary thing. Now I want to go to different parts of the body. Now I want to be contagious. Now I'll go into the blood and the lymphatics and I'll enter different parts of the body. That is very, very important. If you can learn, incubation period is 2 to 10 days. Not very important information, but what I'm going to tell you now is important. Thoda sa common sense laga. What do you think would be your specimen? If there's so much of involvement of the lungs, your specimen will always be bronchial washings or some sputum, and you know everything that I'm talking about up till now is gram-negative Coccobacilli. What are the stains that you will use? Because they are little chota organisms, you will use the silver impregnation stains. Silver matlab what color will you get? You will get a black color. ठीक है? What is the culture media that you will use? You will use the BCYE. What is BCYE? Buffered charcoal yeast extract. Charcoal भी white, black color ही होता है. And serology के लिए you will use ELISA. So why did I say that when you're talking about Legionnaire's disease or you're talking about pneumonia caused by it, it is a black day for the lungs. Black, actually lungs black नहीं हो रहे. Black मतलब एक तो lungs का, lungs anyway black होते हैं because of carbon है ना, तो एक तो lungs का problem है. दूसरा, the stain is also gonna be black and the culture is also gonna have something charcoal black है ना, so there is gonna be the buffered charcoal yeast extract. Finally, how are you going to do the treatment which you are doing for everything up till now? That is with macrolides. So this is also done. Now before I begin with anything new, I want to ask you all something. I'm kind of getting confused. What was this culture media? BCYE. When we say that we are talking about BCYE, you will say, okay, buffered charcoal yeast extract. Ma'am, black kuch bhi hoga, that is going to be for Legionella. Legionella is going to be all that black. If I say the CCFE and the CCYE, ये वाले कहाँ use हुए थे? You said, ma'am, so much of CCC you are saying. This was for, yes, can you recall? Clostridium difficile. This was for Clostridium difficile. That is how we had learned. Clostridium difficile shows you the CCFE and the CCYE. The charcoal wala is going to be shown by the lungs which are going to go black. Finally, I also have one more very similar sounding. There is something known as MIPA and PEMBA. There is something known as MIPA and PEMBA. I gave you a mnemonic for this also. So this I had told you sounds like that very famous reel on that meme that has been made on social media of a very famous politician giving a speech of Rumba Rumba, if you remember. If you've not, 2 years ago, you can go Google it out. Yes, there's a Rumba Rumba speech that has been given by a very, very serious politician who ended up giving a speech called Rumba Rumba. So MIPA PEMBA sounds like Rumba Rumba. This is gonna be Bacillus cereus. So remember, BCY, अगर कोई charcoal black आया तो lungs का black Legionella. CCCC आया तो difficile. And serious politician ने एकदम से बोला, no proper speech, started saying Rumba Rumba, that kind of speech, that was MIPA PEMBA. So now there should not be any confusion. BCY, charcoal for Legionnaires. Now let's move on to the next one. There is an organism that I have taught you earlier, so I've just got you for a very, very quick recap. What is that? Klebsiella granulomatus. Go to the Klebsiella chapter. You have studied it, but what all did I make you read about it? Klebsiella granulomatus earlier was known as Callimatobacterium granulomatus. Now it is Klebsiella. What did I tell you? It causes a disease called granuloma inguinale or donovenosis. Basically, it was causing a genital ulcer. And I had told you that Klebsiella granulomatus is going to cause what kind of an ulcer? It is going to cause a painless ulcer. Yes, I've given you in the, if you go to the spirochetes wala chapter we have done na, painful, painless कौन सा होता है? जल्दी से 30 seconds में revision बताओ. Which are the genital ulcers which are painful, which are painless? So we had done that HSV2, herpes simplex 2, as Haemophilus ducrei, and Haemophilus ducrei where the person will cry with pain. These are the painful ulcers. Whereas when I'm talking about the syphilis में सब कुछ painless था. Then I told you this Klebsiella is also painless, and lymphogranuloma venereum is also going to be a painless ulcer. So these painful and painless genital ulcers, if you're feeling, oh my god, what is ma'am saying all of a sudden, go to spirochetes chapter, pura detail mein we have spoken about it. Okay, so yes, Klebsiella also shows you a painless ulcer, and I told you what is this disease known as donovenosis. So what is the bodies that you see? On a right Giemsa stain, you see the Donovan bodies. Donovan bodies have a very, very famous safety pin appearance. They'll have a staining over here on this pole and a staining over here on this pole. So they will basically look like safety pin appearance, which is also known as what? The bipolar staining, because it has taken up a stain on the two poles. देखो ना, यहाँ पर staining है, यहाँ पर staining है. Bipolar safety pin. The treatment of this is gonna be tetracycline. So this was just because many books have written it under miscellaneous. Jaldi se I thought I'll revise with you. Now coming to the next one, and that is another miscellaneous one that you study, and that is going to be the erysipelothryx. Erysipelothryx is something which has not any feature. Motility nahi hai, sporing nahi hai, capsulation bhi nahi hai, sab kuch no hai. Actually, you just have to know one thing. Erysipelothryx is going to have an erysipeloid. Wow, what a revolution ma'am has done. So Erysipelothryx has erysipeloid. What is erysipeloid? This is something that you're seeing over here, this reddish thing. This is going to be in people who handle any kind of animal products. So animal wale products ho gaye ya fish products ho gaye. So whenever there is any cut or an abrasion and this can come in contact with it, that is what is going to result in this erysipeloid. Please note that the medium is going to be the Tinsdale or the tellurite medium. Where else have you used the Tinsdale and the tellurite medium? That is by Corynebacterium diphtheria, if you remember. So diphtheria maybe there was tellurite and Tinsdale medium, and you have the treatment with penicillin. So remember, especially the story of someone working as a fisherwoman or someone working with the animals, and ye reddish wala erysipelothryx wala region hoga. So when they write next time when you revise it, erysipelothryx, please remember, firstly it is going to result in ER. ER matlab, or ERY you can say, that is going to be, can you see a very reddish color region? That is erythema. Erythema matlab, what is this known as? It is known as erysipeloid. It is known as erysipeloid, right? After that they will say, what is this thrix? You will say T for the tellurite medium, or it is going to be the Tinsdale medium. Tellurite medium or the Tinsdale medium is how you're gonna deal with it. And then Tinsdale ke baad, the treatment is penicillin. Erysipelothryx याद हो गया? This was not a mnemonic I had planned for you. ये बहुत अभी ऐसे impromptu बना है while recording it, so it's good. Erysipelo हो गया, Thrix हो गया for Tinsdale and tellurite, and that is done. Now we go on to another group of quickly one slide wala organisms. There is something known as a gram-negative non-motile family called Hackek. पहले this I've taught you. I have taught you Hackek somewhere because H was Haemophilus. In there I told you that Haemophilus is also a part of a group known as the Hackek bacteria. So Hackek, H for of course Haemophilus, and A for Aggregatibacter, C for Cardiobacterium, E for Eichinella, K for Kingella. Who is the most aggressive out of these? King is the most aggressive. King has to be the most powerful. So who is the most aggressive and the most powerful? Kingella is the most powerful. Next, I taught you that all of them are non-motile, but which is the only one that can show you twitching motility? That is going to be Eichinella. Aikinella says, नहीं मुझे तो थोड़ा सा motility दिखाना है. So you say, does it have a flagella? No, not exactly a flagella, but it has something like a flagella called type 4 pili. So now I can make you revise. Which is the cocci? Cocci तो कभी भी motile नहीं थे, but which is the cocci which has a type 4 flagella, which type 4 pili, which can make it motile, and that cocci is going to be Neisseria. Go back to Neisseria. You have done this over there also. So simple. Out of this Hackett, Haemophilus, Aggregatibacter, Cardiobacterium hominis, Aikinella and Kingella. Kingella is the one that is the most aggressive wala cheese, and Aikinella is the one that is going to show you the twitching motility. Also, Aikinella को बहुत problem है. Aikinella also shows you the needle lickers osteomyelitis. So many people who are IV drug abusers, they tend to lick the needle before injecting it. So needle lickers. So basically many of these Hackett wala organisms are also our normal mount commensals. So when they lick the needle, the organism gets onto the needle, they inject it, and then it can cause osteomyelitis. So it is the needle lickers osteomyelitis, which is by Aikinella. Aikinella is the one that is going to show you twitching motility. But together as a family, where have they been implicated? Together as a family, they are known to cause prosthetic heart valve, endocarditis. They are known to cause prosthetic heart valve endocarditis. Done with Hackett family. Let's move forward. The next one that you have to know is Acinetobacter baumannii. Where have I taught you Acinetobacter baumannii? If you remember, this was belonging, I'll give you the family. There was a family where you had Pseudomonas. Okay, then you had a little bit of Burkholderia, like Burkholderia pseudomeli, Burkholderia malai, Burkholderia cepacia, and in the end of it we had said कि हाँ कुछ miscellaneous सा भी है Acinetobacter baumannii. Basically these were all the non-fermenters, है ना? These were all the non-fermenters that you have. So what is famous about Acinetobacter baumannii? This is in the hospital environments. So the kind of infections also that it's going to do are going to be many kind of nosocomial infections. Hospital wala infections it's going to cause, opportunistic. So it can cause what all? Catheterization has been done. So it causes UTI post-catheterization. Patient is having bloodstream infections, ventilator acquired pneumonia. So all hospital wala infections. That is why do I keep telling you the nosocomial priority list, the nosocomial list that of nosocomial infections that has been given as a priority list for WHO. Which organisms have I been telling you so many times? I don't want to increase the length unnecessarily by telling you again and again. So escape organisms. If you remember escape organisms, I told you I've taught you this under this entire family. Pura detail में पढ़ा हुआ है. So if you remember, Acinetobacter baumannii was a type of this particular escape group that it is very famous for causing nosocomial infections, hospital wala infection. तो कुछ पढ़ना ही नहीं. If I know Acinetobacter baumannii comes under escape organisms, hospital wala infections, तो सारा hospital wala infections होगा, and it is found in the hospital environment. Perfect. Let's move forward. What is the next one that we have to know? The tularemia. We are now left with, actually this is the last one that you have. Tularemia, which is known as Francisella tularensis. तो ये तो कुछ याद हो गया मैम. Tularensis is going to cause tularemia. This is, I always refer to this like plague जैसा infection है. Zoonotic infection है. Zoonotic मतलब it's going to be something that we are going to come in contact with some animal and then get it. Either you get it after touching, contact, or you get it by aerosol inhalation. So होता क्या है? Like I'm saying, plague जैसा है ना. तो plague also you can have either by coming in contact with an animal, or you can get it by inhalation also. There is a pneumonic plague also ना. तो बिल्कुल Francisella tularensis or tularemia is just like very similar to that rodents wala plague. So what are the clinical features that you have? So firstly, there are three forms. There can be an ulceroglandular tularemia. Ulceroglandular means there can be an ulcer, and gland matlab lymphadenopathy. So ulcer and gland, that is ulceroglandular tularemia. After that, what you can have is the going into the lungs, lung involvement, and then you can have an oropharyngeal tularemia. So for example, there is an ulcer in the mouth and a cervical lymph node, ulceroglandular one. After that, you can have an oropharyngeal one, and then you can have a one in the lungs, that is pulmonary. These are the different organs that are affected. Plague was also something you could use as a bioterrorism agent. Francisella tularensis is also something that you can use as a bioterrorism agent. This is also coming under the same. If you ask me, this entire chapter is only gram-negative organism. This is a gram-negative Coccobacillus, yes, but it can be catalase positive. You know, it's not a very common organism. You'll say, ma'am, aapne catalase positive ka list mein you did not tell us Francisella tularensis. It's not a very common organism to know. and hence I did not. But yeah, it can be catalase positive. In terms of culture, you can, but it's very difficult. So best jo hoga, that will be PCR. If you are asked which is the best method, because it's a bioterrorism agent, so you definitely have to know the method for diagnosis, best is gonna be polymerase chain reaction. How are you finally gonna treat it? Treatment is by gentamicin. एक और rare चीज that I want to tell you over here is that it can show you the safety pin appearance. Again, these are some rare things. You'll say, ma'am, na catalase ke list mein tularensis was written, na safety pin bipolar ke list mein tularensis was written. But these are rare things about tularensis that you have to know. That's why I've kept it in the rare most. Let's do a final recap. If I have to ask you about the main features about tularemia or tularensis, first thing that should come to your mind is it is very similar to plague. Plague was a zoonotic disease. This is also a zoonotic disease. Either it could come by contact or it could come by inhalation. What are the most common form? It's going to be the ulceroglandular form, which is ulcer and lymph node. Ulceroglandular form is the most common. If you talk about bioterrorism agent, yes, it is a category A bioterrorism agent. The best test that we have is going to be polymerase chain reaction. The rare things known are it is catalase positive and it can show you the safety pin appearance, and the treatment is going to be, if you want to write it, the treatment is going to be gentamicin. I'm not giving you any mnemonic for this because this is the last bacteria, and I believe that with this short 5-6 bullet points you can definitely learn this. So yes, thank you for being so patient through this entire process of bacteriology. We will definitely be going on to the next section, and nothing is as extensive as bacteriology. I feel this has the maximum number of organisms. So yes, there are a lot of bacteria, virus, parasites, but nothing can beat the bacteriology chapter. I hope a little bit of confidence you got after reading through this.